Fantastic. Thank you very much, Lucy. It wasn't until this morning that I realized there might be some expectations about how awesome this talk might be, and that was a really terrifying thought, so please take any expectations and kick them out the door. I'm in that purgatory right now as a graduate student where I don't have a lot of results, but I have awesome background to share with you. If you were at FameLab last night, then you know that my field site is in the Canadian High Arctic. It's a place where elemental sulfur has been found on the surface of the ice, and is considered as an analog to Europa, and I would also argue also as an analog to Mars. I won't discuss that in this talk, but if you'd like to talk about it, I can discuss those things later over a lot of beers. This is where my field site is located. So in North America, you can see Greenland. There's Ellesmere Island, almost at the top of the world. I thought it was the highest landmass on the planet, but there are two little islands of Greenland that go a little bit further north towards the North Pole. But it's at 81 degrees north. You can see here looking down from the Kármán line at 100 kilometers, the structure of this north-south trending valley. There are mountains on the east and in the west where alpine glaciers are coming down out of the mountain system and forming this large coalescence glacier in the valley. And it's on this glacier where these large deposits of elemental sulfur are found around these sulfide springs. And I'm sorry for not having a scale bar here, but this is a rather large deposit of elemental sulfur seen in 2006, observed that year by satellite through hyperspectral imaging, and also seen in 2007 in the same region. So we can identify this sulfur on the surface from space with satellites. This is a picture that looks kind of terrible now on this screen that I just took a few weeks ago at my field site. Again, you can see this large icing here where there's yellow elemental sulfur on the surface of the glacier. To give you a little scale, this is me standing beside it. I'll go back, so I'll be standing up in the corner up here. It's basically like the same structure standing right there in a little dot. So you can see me standing beside some of this yellow sulfur where it comes off the side of the glacier, fills a small moraine, and goes down through a valley. Here's another look from helicopter when we were flying in and looking down. There's a circular spot up on top of it, kind of like an ellipsoid. That's something we called the blister this year. We've never seen that form before except for this year on the glacier. We don't know why it formed yet. Our current hypothesis is that these things form kind of like lava tubes in the wintertime and form large icings under the snow, deposit elemental sulfur under the snow, and once the snow melts in the summer, we then see all this elemental sulfur, and it's still being formed. Because as you might know from last night, there was a blowout of the one crevasse near me while I was there. When we went back and investigated that region, we watched it over the next few days and saw sulfur forming on top of that fresh snow. And so I have samples of that sulfur now to work with as well. One thing, for anyone who understands the environmental concentrations of hydrogen sulfide, you'll note that this is a really high value. That's the highest amount of sulfide, of hydrogen sulfide, found in any cold spring in North America. To my knowledge, I've never heard of anyone finding any more than that. And so when you're standing there, it smells just like being in Yellowstone. It smells like hydrogen sulfide. And the question originally about this field site is where is all this sulfur coming from? On the left here is a geological map of the valley. So you can see the general structure of the glacier. I have a yellow circle showing where the springs are located. There are only really two large sulfur sources in this region geologically. Most of the rock around the valley is carbonate rock. There's some limestone, some clastics, and some small intervening shales. Shale has sulfur in usually, but not nearly enough sulfur to form the masses of sulfur we found every summer in the Arctic here. By the way, this was first seen in 1988, first observed on foot in 1999, so we think it's been happening every summer for almost 30 years. So anyway, there's two regions in the area, these two geological units in the area that have sulfur in high concentrations, are both anhydrite layers. They contain a lot of calcium sulfate. And so one is the Mount Bailey Formation. It outcrops nearby about 19 kilometers away, but stratigraphically, so in the layers of the rock record, it should pinch out and end long before it gets the 19 kilometers to my valley. The only other culprit then, and the one that we think is really the source of all this sulfur in the form of sulfate, is the Otto Fjord Formation. Outcrops 21 kilometers away, but is extensive in the region. There is diapurism that occurs with the Otto Fjord Formation. That's where, in this case, like a salt would move up through the rock and form large tubes, basically, because it's lighter than the rock around it, and so it moves up. But those diapirs in the region are all 10 kilometers wide and cause a lot of structural variation geologically that we think we would see here in this region if that was happening. So we think the fluids might have to travel 1.5 kilometers into the subsurface to circulate around to take sulfate out and bring that sulfate, that sulfur source, back to the surface. But if there is sulfate, then we have to figure out how we get from sulfate to sulfide, that H2S I told you about. Well, sulfate can be reduced to sulfide in two ways, through thermochemical sulfate reduction or through biological sulfate reduction. In this region, the permafrost is about 500 meters thick. The geotherm, or the change in temperature going down through the rocks, is about 22 degrees Celsius per kilometer, which means that it would take about 7 kilometers into the subsurface for thermochemical sulfate reduction to happen. For anyone who understands geophysics, that's way too deep. At that point, the rocks are pushing too hard for water to get into the subsurface. So what we think is going on is biological sulfate reduction. It makes sense as a pathway, and it also is supported by isotopic data. I'm glad Bradley gave you an introduction to isotopes and what delta means, so I don't have to explain this. For delta S, this is delta 34 sulfur. It's measuring the 34 sulfur versus 32 sulfur. In this figure, you can see the Otto Fjord anhydrite, again, is our culprit. Gypsum precipitate. Gypsum is calcium sulfate dihydrate, so it's remnant sulfate in the spring water that's coming out and forming precipitates on the surface. Appears to be enriched in 34 sulfur, whereas the sulfide and the sulfur coming out themselves are much lower, or depleted in 34 sulfur, suggesting that microbes are taking this anhydrite and are reducing the sulfate so that this is light and this is heavy. And this is supported by another feature in the region that I haven't talked about a lot to anyone here at the conference yet, that we've called paleopipes. Some people in the audience who do paleo work won't like me using this word to describe these things. In this case, these pipes are at most 7,500 years old, so not millions or billions. But these are alteration features we found in the region near the toe of the glacier that appear to possibly be past pipes where fluids had flowed up through the subsurface and contacted the glacier. And when the glacier moved away, these pipes were left sitting out in the environment. And so when we see these structures, there's the carbonate rock out around. That carbonate rock starts to mix with gypsum, and we see iron oxides, iron oxyhydroxides around in that red rim you might see around the feature in the top left there. In the center, it's a lot of gypsum, some elemental sulfur, and some sulfide grains. So it fits the model that we proposed for how this is actually forming. And that supports the overall model of the system. So meltwater is coming off the glaciers nearby, these alpine glaciers, other coalescence glaciers. It's going into the subsurface, potentially taking some of this Permian shale you see here, taking some organic carbon out of that and using that to metabolize the sulfate down below in the subsurface, driving microbial sulfate reduction. And so where my part of the research now comes in is really at looking at what happens on the surface, where there's this oxidation of hydrogen sulfide to form that yellow elemental sulfur you've seen in these pictures on the ice. You can see it there on the left in 2006, a picture from 2007, the yellow sulfur on the ice. When we're considering whether or not microorganisms could be responsible for some of that oxidation of hydrogen sulfide to sulfur, my past lab mate, Catherine Wright, who's been to AbGradCon before moving on, did some work studying genomics of a spring deposit, studying at BF 0901 on the far left there is right at the spring source. And as you move right along this scale, it's going further and further away from the spring. So trying to see what organisms are present. And what she found is that these two organisms, Burkholderia and Ralstonia, appear to be present in the fluids where they emerge from the glacier, potentially being involved in that sulfate reduction in the subsurface. But what's interesting is that this other organism, Flavobacterium, appears to dominate that surface, that veneer of sulfur that's all over this area in tens of square kilometers across my field site. In the past, in the lab, Dovnik Gleason, who was in our lab for a while, was doing a lot of gradient tube culture studies that I continued on, taking gradients of sulfide in the bottom of a plug in a culture tube, allowing oxygen to diffuse in, and the two will mix, forming a gradient of sulfide and oxygen to allow these organisms to grow. And what we found is not only can they form large blooms of elemental sulfur in the center of these tubes compared to controls, but that the structure of some of the filaments and sheaths and other things that are formed are really intriguing and potentially really useful biominerals for studying these processes if they're occurring in the field. I have on this figure on the left is a general EH-pH diagram. It's studying the potential energy being shifted through the environment and the acidity of the environment, given parameters from my field site. And if you look, the general region of the spring waters coming out shouldn't be forming that sulfur rhombus up at the top, the elemental sulfur. It should all be going straight from sulfide up to gypsum in my field site. So we think not only are organisms involved in this process, but they're doing something to stabilize the elemental sulfur at the surface. Once that snow melts and it's present, it really should be oxidized further to sulfate. And we don't know exactly why yet. So in my work, I'll be working to try to figure out if there's various forms of morphology, things I can look at, doing some chemistry to try to pick out polysulfides, which is a mixture of elemental sulfur and one sulfide that are mixed together and are usually produced by microorganisms that oxidize hydrogen sulfide or sulfide. And also, organics are present in the spring fluids, and so it's worth looking into whether or not there's organics around the sulfur that we see. This is an SEM image that I have on the right here of that paleopipe feature. I took some material from it, looked at it under the electron microscope, and although this structure is much smaller than what I saw in my gradient tube culture there on the left, you can still see this long, spindly, filamentous structure that was formed by microorganisms in my samples. I don't have a lot of time to jump through the rest of this stuff I'm going to show you real quick, but I'm also doing a lot of X-ray spectroscopy, X-ray fluorescence studies of the samples I have. This is an image on the right of an X-ray fluorescence map conducted at 2,500 electron volts of my sample. Every atom absorbs X-ray energy at a certain energy, and so I can go in and use that to look at my samples and figure out what kind of sulfur is present in my samples. I'm just going to skip through a couple of these real quick. One interesting thing I found are these things I'm calling sulfur turds. In my sample, you can see it there on the left and some images here on the right that are pure elemental sulfur inside of this sample, but that appear to be surrounded by what looks like an organosulfur through X-ray spectroscopy. And you can kind of see here that this spot 12 map at the top is elemental sulfur, and down here is the stuff that could be organosulfur surrounding it. And so with that, I'd like to show my collaborators. Steve Grasby is my main field collaborator. He's the guy sleeping on the ice right there. And I'll acknowledge them as well. Alexis Templeton's my advisor. She's fantastic. And John Spear and Chris Trivedi are doing a lot of the genomics work, metagenomics and RNA work on all the samples that we have. And again, Steve Grasby is just the man in the field. So thank you.
Questions, anyone?
So your field site is obviously a cold environment, and I'm guessing that you go there in the summer. I'm wondering if these microorganisms that are sulfuring they're cycling the sulfur, if they're freeze tolerant, and if when it gets cold, do they just go into hibernation, or can they still grow? Is there still growth?
I actually haven't studied that, and since I presented earlier this week about doing some freeze tolerant stuff with eukarya, maybe it'd be worth looking into, but I actually don't know. We have them in really cold cultures, and so they're fine, but as far as what their freeze tolerance level is in sub-zero, I'm not really certain. They're easy to bring back once they're frozen, though, so it should be okay to freeze them lower. Maybe I should do a study with that, too. So.
Got time for one more question, if anyone has one? No, well, I have a question.
Please.
I would like to know these groups of organisms you're seeing, we're seeing single species domination or genera?
It's definitely genera, we think. So the Flavobacterium, we've been trying to work in splitting out which Flavobacteria they are, and we have one specific one that Catherine's been working on a lot to try, well, she was working on a lot, that she thinks was the primary culprit for this process. So.
Let's all thank Graham one more time.
Thank you.